Good afternoon, all. Welcome, welcome. This is our practice test session based on yesterday's webinar. So welcome back to those of you that were here yesterday. And if you weren't, never fear. Even if you weren't here for the content yesterday, this can help you figure out where your strengths and your weaknesses are. And our topic in biology today is genetics, protein synthesis, and Mendel's laws. That's what we went over yesterday. And as we go through our practice test, I will go over what question or what response was correct and why it was correct and what's wrong with the other responses. So even if you missed yesterday, you'll certainly get a lot out of that. And if you were here yesterday, this will be a great testament to how much you retain, where your strengths lie, and where you need to study a little bit more. And with that idea in mind, I do want to take us out right away before we get too deep into our plan and fulfill what I had promised you. Yesterday, I went over our question bank because our question bank gets rave reviews from so many students. And I promised you that today I would go over our on-demand videos. Now, our on-demand videos would be something that when you get done with today's webinar and you realize, oh, I really need to work on protein synthesis, or I really need to work on Punnett squares, you could utilize our on-demand videos to make sense of those concepts. So I'm on our website. Notice I'm in the on-demand videos section. I'm going to go into science since that's what we're working on. I'm going to go into biology, life science, and I'm going to pick a particular video such as cell structure and function. And there are a couple of things I want to show you about this video. First and foremost, it's 39 minutes long, but let's say that you only really need a refresher on the levels of organization. They're all time-stamped. You can go right to that topic. And notice that topic is only 20, even though the video, if you want to dive deep for 39 minutes, you can. But if you just need a particular topic, you can go just that topic, and they're not very long. Now, the other thing is I want to eat products that come with it because spending. Let's see what happens here. I'm going to refresh it. Huh. Never had this happen before. Here we go. Here we go. Okay. All is well. Yay. All right. So I said I wanted to show you what comes with these videos because they are pretty spectacular. So we get a cheat sheet with each video and we get notes with each video. Let's take a look at our cheat sheet. Check that out. Is that not a thing of beauty? I love our cheat sheets. I just think these are amazing. This is something that you could, if you had the ability to print, you would download and print, you could screenshot and study on your phone. You have so many options with our cheat sheets. They're packed full of information, they're colorful, they're informative, and most of them even have practice questions with the key upside down at the bottom. So I love sharing the cheat sheets. I think those are pretty spectacular. The other thing that comes with our on-demand videos is our handouts. So this is something I've heard from students. They like to print these, and then when the video annotates, they annotate with the video. Because I'm sure you've learned by now that the more senses you can activate when you're studying, the better you remember. So if you have the ability to print them out and you can circle things, you can draw things, you can annotate as the educator is annotating, it helps you remember it better. Okay, that's just kind of a fact of cognitive learning. But I love sharing all this with you because I just think I'm so proud to work for a company that has so many amazing resources to help you pass your TEAS. That gets me really excited. So in speaking of those resources, as we said, yesterday we went over our question bank. It has over 2,000 questions with their rationales like we're going to go over today. The on-demand videos we just talked about. We also, as you know, offer tutoring. I'm going to skip to, there's your tutors. So I am Shellie Harshberger. I am your science expert. So I can handle your chemistry, your biology, your anatomy and physiology, your scientific reasoning. Tammy can handle all of the English and reading for you, and Brandon, the math. So you can scan those QR codes and reach out for a tutoring session. We'd love, love, love to work with you. As you know, we have these free live webinars. Currently, our format is content one day and practice test the next in the science realm. And I think reading and English is doing that right now too, but we always change that up. Next month, it'll be something different. You can rewatch old webinars. You can use these QR codes to access all this stuff. And you can also, if you're looking to get some of the products, you can bundle them in a SurePass or a SurePass Pro. Now, you may be asking me, well, what's the difference? Well, SurePass has all those wonderful features on our website, the Q Bank, the on-demand videos, the notes, the cheat sheets. The SurePass Pro has all of those things, plus an hour of private one-on-one tutoring. So just whichever you feel you need to get you to success for the TEAS, that would be the package you would choose. Now, if you haven't been following us on socials, I strongly encourage that you do. Each week, we focus on a topic, and then we do 2 mini lessons and 2 practice questions. And you can see those for free on TikTok, Instagram, Facebook. And it's free practice for that TEAS you've got coming up. Also, if you're thinking of tutoring and you check our website and you're like, oh, none of those times fit my schedule, certainly email us at prenursing@archerreview.com. Sometimes we can move our schedules around and accommodate you. So it's certainly worth the ask. Now, before we dive in today to our practice test, I do want to just give you some heads up. People who have signed up and who are coming at me from Zoom, when we go through the practice test, if you put your answer in the practice, in the chat window, I am the only one that can see that. That gives you a little bit of confidentiality, a little bit of comfort to throw an answer out there, maybe if you're not 100% sure. If you're coming at me from Instagram, YouTube, Facebook, any of the socials, you don't have that confidentiality, but I still strongly encourage you to participate. Take a risk, even if you're not 100% sure. Everybody learns, and everybody learns from their mistakes. And when you throw that response out there, again, you're activating another sense, and that will help you remember. Even if you get it wrong, it'll then help you remember when you're taking the test. Oh, wait, that was the one I got wrong. What was the correct answer again? It helps bring that to mind. So please participate. Please throw your answers out into the chat box, and we'll see where it goes. Now, also, this is a timed webinar. Each question will be timed for 45 seconds, and that helps mimic your actual practice, or helps mimic your actual TEAS exam experience. So I will read the question. I will not read through the answers. Some wonderful feedback I got said that's more distracting. So I will read through the question quickly. I'll activate that timer. And then when it goes off, we'll reveal our responses and we'll talk about why the correct answer is what it is and the incorrect answers are incorrect. All right, so our first category is genetic material and protein. What is the correct relationship between DNA, chromosomes, and genes? Select all that apply. All right, let's take a look. Let's see how we did. Correct answers are A, C, and D. Now let's go back and break those down. A, DNA is packaged into chromosomes. Yes, remember we talked about how DNA coils up tightly around histone proteins into chromosomes. And that also then explains why D is correct. So I'm going to jump down to D. Chromosomes are made of DNA and histone proteins. Yes, DNA coils around those histone proteins and they help organize the DNA. All right, our other correct answer was C. A gene is a section of DNA that codes for a protein. Remember from our content yesterday that there are genes all over the chromosomes, and a gene represents a protein, which represents our traits. Brown hair, blue eyes, things like that. Now, let's break down why B and E are incorrect. B, genes are long stretches of chromosomes. What's wrong with B is this word long. Genes are small segments on the chromosomes because chromosomes hold the instructions for many, many proteins and many, many traits. And so that's what makes B incorrect. Now let's take a look at E. DNA and chromosomes are unrelated structures. I think everybody probably knew that that's not accurate. DNA is what makes up chromosomes. And so that makes that an inaccurate statement. Wow, that was hard to say. All right, folks, we're going to move on to our next one. In the structure of DNA, where are the sugar and phosphate groups located? All right, I did see some correct answers out there. Let's take a look. Correct answer is C, along the backbone of the DNA molecule. Now, I know I had in previous webinars, someone asked me, what do we mean by backbone? We could say backbone or we could say sides of the ladder. Let's take a look. I've got a visual on our next screen here. So if you recall from our content yesterday, the sides of the ladder or the backbone of the ladder right here is where the phosphate and the sugar part of the nucleotide reside. Here's our phosphate, here's our sugar. Okay, now let's look at our other answer responses. A, in the base pair, excuse me. A, in the base pairs at the center of the double helix. So if we look at our illustration, these are our base pairs. They are nitrogen bases. They are not composed of phosphate or sugar. And so that's what makes A incorrect. B, in the rungs of the DNA ladder. Remember, sorry guys, I'm struggling a little bit. In the rungs of the ladder, remember that I said that the rungs are also known as the steps of the ladder. And again, the steps are the base pairs, not the sugar and phosphate. So that makes that incorrect. Looking at D, inside the ribosome. Sorry, that's just silliness. The ribosome is where protein is made, and DNA can't leave the nucleus. So that makes that very incorrect. All right, let's take a look at our next one. Which of the following are components of a DNA nucleotide? Select all that apply. All right, let's take a look. A, B, and D. A, phosphate group. B, deoxyribose sugar. D, nitrogenous base. Let's take a look at the illustration I have on the next screen. There's our phosphate group. There's our deoxyribose sugar. And here is a representation of one of the bases. Remember, the bases are A, T, C, G. Now let's take a look at our other responses. Where are those inaccurate? Oh, and I'm sorry, I said ribose sugar, but I checked. C, that's incorrect. My bad, folks. Sorry, I'm still struggling with my coughing here, not trying to cough in your face. So let's put it in the right spot. There's our nitrogenous base. So now let's break down what's wrong with C and E. C, ribose sugar, is the type of sugar that's in RNA, not DNA, RNA. And so that's what makes that one inaccurate. And then E, amino acid. Amino acid is the building block of proteins. It is not part of a DNA nucleic acid. All right, moving right along. Let's take a look at our next question. What is a key feature of RNA that differentiates it from DNA? Our correct answer is C. How'd you do? Let's take a look. RNA contains uracil instead of thymine. That is accurate. Okay, let's break down our others. RNA is double-stranded. No, RNA is single-stranded. It is just one half of that spiral, like DNA, but it's one half of that. So that makes that incorrect because of being double. B, RNA contains deoxyribose. No, RNA contains ribose. It only contains ribose sugar, not deoxyribose. And that's where its name comes from. It's ribonucleic acid. DNA is deoxyribonucleic acid. So the name gives it away. The sugar is the first letter, the ribose part of it. Then D, RNA remains in the nucleus. The whole job of RNA is to take the instructions that are in DNA out to the ribosome in order to build a protein. So if it remained in the nucleus, it would not be doing its job. So that makes that inaccurate as well. All right, moving right along, let's take a look at our next one. Which of the following can cause genetic mutations? Select all that apply. So I had someone tell me that the select all that apply really challenges their brain. And I agree with you on that one. But when you have these on the TEAS, if you get, let's say there are 3 correct answers, if you get 2 of the 3, you don't get partial credit. So it's really good to practice them and challenge your brain despite how painful it might be. All right, are we ready for our reveal? A, B, and E. Let's take a look. A, errors during DNA replication. That is one of the primary ways that a mutation can occur. Now, granted, there are checks and balances that might fix that mutation, but not all of them get fixed. All right. B, exposure to environmental factors such as radiation or chemicals. Yes, that is another way that a genetic mutation can occur. Then one I don't believe I really talked about in great detail yesterday, but it kind of falls also under environmental factors is E, viruses. Viruses that insert their genetic material into host DNA. So it's actually really fascinating. If you do any research, our entire DNA as the human genome is like 8% viral DNA. DNA from viruses that injected itself in our ancestors, and then that got passed down generation from generation from generation. And in some cases, they're actually protective. There is a viral insertion, a viral part of the DNA in some people in Africa that will prevent them from getting HIV. And it's because of this viral DNA that's inserted in their genetic makeup. So it's again, if you guys have heard me talk about viruses in the past, you know that I find them terrifying and amazingly fascinating all in one. And that's one example of why I find them so fascinating. All right, let's break down what's wrong with the other responses. C, proper functioning of DNA repair mechanisms. Well, if it's proper functioning, then we're not going to have a genetic mistake, which is a genetic mutation. Mutations are mistakes. And so if we have proper functioning going on, then we're not going to have a mistake. So that makes that inaccurate. Now let's take a look at D, the correct pairing of nucleotides during replication. Again, if things are working well, then we're not going to get that mistake of a mutation. So that makes that incorrect as well. All right, our next one. What is the function of a gene? I bet everybody got this one right. The correct answer is C. C, to carry instructions for making proteins. So remember, genes are a segment of chromosome, and they hold that recipe for making proteins, and it's proteins that determine our traits. Now, let's take a look at the other responses. A, to bind with ribosomes. I'm not even sure where they were going with this one. Maybe that's talking about mRNA and how mRNA lays out on the ribosome when we have protein synthesis. That's possible. B, to digest proteins. Digesting proteins or breaking down proteins would be the job of like lysosomes within the cell and would not be a function of the gene. Then we have D, to mutate DNA sequences. No, again, we learned in our last slide and yesterday that mutations are mistakes, and a gene is certainly not something that's going to foster a mistake on purpose, at least. So we will consider that incorrect as well and take a look at our next one. Which of the following statements correctly describe transcription? Select all that apply. How we doing out there? How we feeling? Let's take a look. Let's see what our correct answers were. B, C, and E. Let's break them down. B, RNA polymerase is the enzyme involved in mRNA synthesis. Remember that transcription is our first step in the process of building a protein, and it involves RNA polymerase essentially making a photocopy of the instructions of the protein that we need. All right, our next one, C, mRNA is complementary to the DNA template strand. So remember that mRNA is essentially making a photocopy of that segment of DNA that it needs to make the protein. But when they say it's complementary, it's because they're matching the bases. They're matching. If DNA had an A, RNA would be matching a U with it. If DNA had a G, RNA would be matching a C to it. So they call it complementary because how it's making its so-called photocopy is by matching the complementary base, the base that pairs with what it's reading. Okay? And then E, the first step is unwinding the DNA. So yes, remember that DNA is a spiral double helix. And so we unwind the DNA and then RNA polymerase works to build that photocopy or that mRNA strand. Which of the following statements accurately describe translation? Select all that apply. Let's see our answers. There are 4 correct ones here, B, C, D, and E. Let's break them down. B, tRNA aligns its anticodon with mRNA's codon. So remember that mRNA is read in groups of 3. That's a codon, and it's like 3-letter words. And how tRNA knows it's bringing the right amino acid to build that protein is it lines up its anticodon with the mRNA codon. So for example, if the mRNA codon was AUG, the tRNA codon would be UAC. And the tRNA then knows, I've got the right amino acids. These line up. And so it can drop that amino acid off and go back for another. Now, C, each tRNA carries a specific amino acid. Yes, it carries the specific amino acid that relates to AUG or to whatever codon it's dropped or whatever codon it's lining up with to drop that amino acid off. D, translation occurs in the cytoplasm on the ribosome. Yes, remember that translation is the second step in protein synthesis. It's where the protein's being built. E, the end product of translation is a protein. Yes, it is a polypeptide chain that will ultimately go out to other organelle to get folded into a functional shape, but it is still a protein when we're done with translation. Now, what's wrong with A? Translation directly converts DNA into RNA. No, translation is converting mRNA's message into a protein, and that's what's wrong with A. Okay, well done. We're through the first part. Now we're on to Mendel's laws. Now, remember there were 3 Mendel's laws. And so ultimately, when we have our next three questions, I can't, excuse me, I can't go over the other answers because they kind of give stuff away. But once we get to the third one, I'll go over all the answers. And I think that will make it very clear about Mendel's three laws and how they are different from one another and what they mean. Let's take a look. Which statement best describes Mendel's law of dominance? Ready for the reveal? Here we go. C. How'd you do? Let's take a look. A dominant allele will be expressed in an individual if at least one copy is present, while a recessive allele is only expressed if both copies are recessive. So be attentive to the fact that the law of dominance talks about dominant allele expression, and that can help you put the name of the law with the actual explanation of it. Let's take a look at our next one. What does Mendel's law say? What does Mendel's law of segregation state about alleles? Here we go. A. Correct answer is A. Each organism has two alleles for a trait. Remember, I said I kind of think of them as like the right and left shoe. They're both the same pair of shoes, but they're slightly different. But they separate during gamete formation. Remember, those are sex cells, so that each gamete carries only one allele. And remember, that ties in with meiosis. We talked about this a little bit yesterday, where we have a parent cell of 46 cells, but the daughter cells, the 4 daughter cells in meiosis, whether they're eggs or sperm, have to have 23 chromosomes, so that when a sperm with 23 meets with an egg of 23, we have the right chromosome number for the zygote. And so this ties in with that. This is telling us then that on those alleles, there's genetic information that's coming from the sperm dad, the sperm donor dad, and the egg donor mom. And finally, which of the following best explains Mendel's law of independent assortment? Our correct answer is A. Let's take a look. Traits are inherited separately, meaning the inheritance of one trait does not affect the inheritance of another trait. So what that means is If you inherit mom's brown hair, it does not necessarily mean you're going to inherit her blue eyes. They don't influence one another. Okay, that's what that's saying. So the law of independent assortment means that each trait is inherited independently, meaning without influence on other traits. So that makes that the correct answer. Now that we've went through all three, let's take a look at the other answer choices. B, some traits such as hair color and eye color must be inherited together. Now that is not correct as far as Mendel's law of independent assortment. I can say, though, that sometimes there are genes with traits that are so close together that when they do crossing over in meiosis, they do have a greater probability of being inherited together. But they don't influence one another, and so that still aligns with Mendel's law. All right, C, alleles for a single trait separate during gamete formation, ensuring each gamete gets only one allele. That's the law of segregation. And that's why I couldn't go through the first two, all of their answer responses, because they all had the different laws embedded in there. So C is the law of segregation, not the law of independent assortment. D, a recessive allele will always be masked by a dominant allele when both are present. That's the law of dominance. And so again, I couldn't go through that earlier. I would have been giving it away before we got through all three. Okay, let's see what our next question is. And let me get our timer out of the way. In a monohybrid cross between two heterozygous individuals, capital F, lowercase f, crossed with capital F, lowercase f, what is the expected phenotypic ratio of the offspring? Be attentive to this word right here, and we're going to get the timer going. Now, before we go through the answer, or before we reveal the answer, I should say, let's go through this one. Let's talk about how to do this. So if you have a heterozygous cross, capital F, lowercase f, and capital F, lowercase f, so that represents the potential alleles from one parent and the potential alleles from the other, we set it up like this. That represents one of them. That could potentially be a possible sperm, another possible sperm from that heterozygous male. Here, that could be a possible egg from the female. That could be the possible egg from the female. Then we pull them into the Punnett square. See how that capital F came down, this capital F came over. Capital F came over, lowercase f came down. Do the same thing in the bottom two squares. And now we have to make sense of it. So we're looking at the genotype and the phenotype. And remember, the phenotype is what the question asks for. So the genotype, we just look at what's in our square. We have one dominant, homozygous dominant, capital F, capital F. We have two heterozygous and one homozygous recessive. But remember, this is where this is where Mendel's law of dominance comes in, because phenotypically our numbers are going to be different, because all three of these are going to show those freckles, the dominant trait, okay, because they have at least one F in their pairing. So in that case, we have 3 out of 4 dominant, 1 out of 4 recessive, or we can say a 75% chance of getting that dominant trait and a 25% chance of getting the recessive trait. Now, as I said yesterday, remember that these are probabilities. This does not mean that if we had 4 kids, 1 would hold the dominant, homozygous dominant, 2 would be heterozygous, and 1 would be homozygous recessive. That does not mean that. This is a probability situation. So, given all that information, that means that answer B was correct. Our ratio was 3 dominant to 1 recessive. Okay, so what's wrong with the others? Oops, went too far. 1 dominant to 1 recessive, that doesn't line up at all. As a matter of fact, since it's a ratio and it's a Punnett square with 4 squares in it, we would want our numbers to add up to 4. So that makes that incorrect. B was correct. C, 2 dominant and 2 recessive. We did not see that in our cross. We saw 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive, which yielded 3 dominant appearances in phenotype and 1 recessive appearance in the phenotype. D, 1 dominant, 2 heterozygous, 1 recessive. I would say if anybody made a mistake, it was probably D, because you were more than likely reporting the genotype. You were more than likely reporting this instead of this. And that's where that may have went wrong. And so that's why it's so important to really read the question closely and determine, is it asking for genotype? Is it asking for this, or is it asking for this? Okay, let's take a look at our next question. Which of the following correctly differentiates incomplete dominance from codominance? Let's see how we did. Our correct answer is C. In incomplete dominance, the heterozygous phenotype is a blend of the 2 alleles, while in codominance, both alleles are expressed simultaneously. And I'm going to go back to our other slide because I have our visual from yesterday. So remember that in the cross, or in incomplete dominance, if we looked at snapdragon flowers, if we took a white flower and crossed it with a red flower, all of the offspring would be pink. That's incomplete dominance. It's like mixing white and red paint together. Whereas codominance is like co-workers. They both show up simultaneously, as in our checkered chicken. Okay, let's take a look at the other responses. A, in incomplete dominance, both alleles are fully expressed. That means show up. While in codominance, the traits blend together. And you can see that they just crisscrossed them. If they flipped these two and put codominance up there and incomplete down there, it would be correct. But in its current form, it's incorrect. B, in incomplete dominance, one allele completely masks the other, while in codominance, only the dominant allele is expressed. So this is kind of mixing some of Mendel's genetics with non-Mendelian genetics, and none of it is accurate. If you recall from yesterday, Mendel just believed, as we learned about the laws, there are dominant traits or there are recessive traits. There's no in between. But non-Mendelian genetics, genetics that we now understand at a more complex rate because we now understand DNA, tells us that's not always the case, that some genes have different types of inheritance, like codominance and incomplete dominance. And so we don't have this idea of them completely masking one another in non-Mendelian genetics. Let's take a look at D. In incomplete dominance, the recessive allele is never expressed, while in codominant, the recessive allele is only expressed if 2 copies are present. And again, that's mixing non-Mendelian genetics, this idea of incomplete dominance, with Mendel in the rest of the explanation. And that is inaccurate as well. All right, let's take a look at our next one. Why do males have a higher probability of inheriting X-linked genetic conditions compared to females? Our correct answer is B. Males inherit only one X chromosome, so they do not have a second X to mask the recessive allele. Let's take a look at how that works. So let's say that we have a female, and she's what is known as a carrier because she has a homozygous dominant, or she has a dominant trait and she has a recessive trait. So she's heterozygous, and they call that a carrier. And we have a male who just has that one X chromosome, and he has the dominant R on there. Okay, now let's take a look how we do this in a Punnett square to show the probabilities of male versus female in this scenario. So there's our female with her superscript R. There's her X with our another superscript lowercase r. So this top is representing our female. There's our male along the side. Notice how we bring them in, keeping that superscript. Okay, so a couple things to draw to your attention here. First of all, it's males who determine the sex of offspring because they're the only one who can donate the Y to make it a boy. And second of all, notice since they don't have that other X, they don't have anything to cover up having a lowercase letter, this recessive trait. They don't have another X to cover it up. Notice how she up here has this R over here that can cover up this recessive trait. So let's look at how we report this, because we have to report it differently since the outcome is different for males versus female. So in this case, we say we have one female that is unaffected with the condition. What do I mean by unaffected? It means she's not a carrier. She doesn't have the trait in her genes whatsoever. So we have a 1 out of 2 probability here, because we're only looking at the females, of being unaffected. And that's what we're looking at here. Then we say we have one female that's a carrier right here. So she carries it in her genes, but she doesn't have the condition. Then we have one male unaffected and one male affected. So genotypically looks pretty similar, but here's where it changes. Phenotypically, now we have to look at it as just females and just males. In females, 100% would not express the condition. 50% would be carriers, but 100% would not express this condition. But look at the males. 50% of them would have this condition. So the probability is different, whether it's male or female, when it comes to an X-linked disease or an X-linked condition. All right, folks, you guys have been great, phenomenal, awesome. Thank you for being here. I always like to bring this page back up just in case you missed it when we first started. Remember, if you have an iPhone, you can take a screenshot and all of those QR codes will be active. If you have an Android, you could still take the screenshot and then you would want to punch in those those internet addresses. Also, remember, we've got bundles, so we can save you a little bit of money by packaging the products together depending on your needs. SurePass Pro has everything with an hour of private tutoring. SurePass just doesn't have that private tutoring. Speaking of tutoring, there we are. There are your tutors. I'm Shellie. We got Tammy for English and Reading, Brandon for Math. Use those QR codes or reach out to us at archerreview.link/TEAS tutors in order to set up an appointment. Last but not least, if you could please scan that QR code and give me some feedback. I listen to it. I follow it. I'm happy when I see it. So let me know how we did. Let me know how I did. And just an FYI, if you're looking for another free webinar, I'm coming at you again Thursday and Friday of this week. So tomorrow will be scientific reasonings content, and Friday will be the practice test. Same time, 3 p.m. Eastern Standard Time on both days. And I'd love to see you there. So I'm going to go ahead and mute this and leave our QR code up just for a little bit for the feedback. But otherwise, you guys have a fantastic day. And I will look forward to seeing you hopefully tomorrow. Take care.